So RNA interference. RNA interference is the process by which double-stranded RNA induces sequence-specific gene silencing by targeting mRNA for degradation. The process blocks and silences gene expression, and thus leading to a loss of function of that specific gene or a knockdown. Um, it is also used to study cellular functions of that gene, which is very important for cellular biology. Um, and now we'll go through how the method works. I have a diagram also of that method. Awesome. Double-strand RNA is introduced to the cell. It then triggers the RNAi pathway of cells. So it recruits the dicer, which is an enzyme. Um, the dicer cleaves the double-stranded RNA into smaller fragments, about 20 to 25 base pairs long. And then RISC distinguishes between sense and antisense strands. And what it does, RISC attaches to the antisense, and the sense strand is degraded within the area. The RISC pairs up with an mRNA and attaches to the specific targeted complementary base pairs. And then it cleaves the mRNA and stops protein synthesis. And it results in that gene being silenced or loss of functions of that gene. And then we can just check all the methods one more time. Right. So pretty much all I said is how it works right here. And yeah.
Very nice. And take it away, Gabby.
Great, thank you. So before we delve into more of the specifics of the paper that we chose for our project, I thought of a helpful analogy that I could present to you all that might help grasp the concept a little bit more. So when I was first reading about the technique, what came to my mind was sort of just a light switch. But more specifically though, since there's been some evidence of our method having some flaws in it, I compared it to a dimmable light switch in the bedroom that I share with my sister at home. So in this analogy, the light switch is our section of mRNA where its expressed product, quote unquote, is light. And my family comprises the long strand DNA. Next, the car that I take to drive from my family home to my college home works in a similar way that the dicer protein functions to break up the DNA into smaller fragments, sort of, in a way, breaking up my family unit. Then once I get to my college living space, I sort of discard my car like the dicer protein gets discarded since I don't need to drive around as much. And instead of being attached to my car, Cal Poly takes me under its wings in the way that the RISC complex takes a new, smaller nucleotide segment. And now that I have more control over the brightness of my bedroom, since I'm not sharing the space with my sister, I can fully control the expression of brightness in my bedroom. So in the analogy that I'm creating here for you all, the RISC complex is sort of just compared to me as a person. I'm the thing that's acting on the mRNA strand, aka this light switch, to affect it. And unless some other person turns the brightness down in a room, it's going to stay on the highest brightness I last set it to. And once I attach the mRNA, or once I reach the light switch to make the room dark, I will effectively destroy or affect the expression of light. And now with that, I will talk a little bit about the paper that we chose. So we based our whole video off of an article that's called Gene Knockdown in Malaria Parasites via Non-Canonical RNAi that was published in 2019. Their main question was, can this method be used to treat pathogens such as malaria? Malaria is an aggressive parasitic disease that a lot of scientists have sought out to find a cure for, but to not much avail. So the Henschel group realized that an innovative method like RNA interference to work towards finding a therapeutic agent for the disease wouldn't really be possible. And that's just because the host of the disease is an organism which isn't equipped with the appropriate machinery that the RNAi requires. More specifically though, the species doesn't have the dicer enzyme and the argonaut protein necessary for the method. And while my group mates are going to go in greater depth about this, essentially the dicer enzyme slices up the double-stranded DNA, and the argonaut protein attaches to the RISC protein, which sort of activates it and helps for proper degradation of the mRNA. Lastly, the group just hypothesized that an artificially created RNAi system would be sufficient to study the species in order to get a better understanding of what would be needed to create an efficient anti-malarial drug.
All right, so now we're going to move into how the method addresses the question of the paper. So given our understanding of RNAi knockdown and the fact that the Plasmodium genus lacks the necessary cellular machinery to carry out canonical RNAi knockdown, no dicer protein present, can a certain method of RNAi be constructed to allow for treatment of pathogens that fall under the Plasmodium genus? So dicer, as we discussed, typically processes transcribed shRNA into siRNA, which is then used for degradation of mRNA, ultimately inhibiting translation. Given that no dicer protein is present in malarial cells, the authors investigated a way to manufacture an exogenous strand of RNA that would result in mRNA degradation. An alternate pathway has been discovered in mammalian cells using AGO2, what Gabby previously mentioned, and this processes the shRNA. And AGO2 stands for Argonaut RISC Catalytic Component 2, and it's a gene that, when translated, binds to the RISC protein, inducing degradation of mRNA. So these AGO shRNAs serve as the key to the RNAi pathway being tested. And tests were done on P. burgii, the rodent equivalent of malaria in humans, and a GFP transgene in P. burgii that was stably and ubiquitously expressed was chosen for continued experimentation within mice. Moving on to results. The first set of results assessed the GFP knockdown efficiency in wild type versus dicer deficient MEFs, or mouse embryonic fibroblasts. The anti-GFP AGO shRNAs, along with the positive control, reduced GFP fluorescence significantly more than the wild type cells compared to the negative control. Conventional shRNA also failed to reduce GFP translation in dicer deficient cells. However, all 4 anti-GFP AGO shRNAs that were tested showed successful inhibition of GFP expression in dicer deficient cells. And for strain number 2 in the chart, GFP expression was reduced to about 50%. And so the authors furthered their investigation by exploring the effectiveness of their new method of RNAi knockdown in P. burgii in different stages of the parasite life cycle. So we have here the negative control maintains less GFP inhibition than the AGO shRNAs in all tested stages. And the mRNAi knockdown using AGO shRNAs was deemed to be possible throughout several stages of the parasite's life cycle. Viewing the chart, we see we have the oocytes, sporocytes, and the later stage, 48 hours. In all cases, the anti-GFP AGO shRNAs inhibited the GFP transgene more than the controls. And the authors concluded that they have successfully constructed an alternate RNAi pathway via these AGO shRNAs. Here are our sources, and thank you for listening.